Hi, my name is Karina Ghaffari, and it is 8/28/2020, and I will be discussing the determination of gender, genes versus hormones. Sexual development is programmed in the human genome. Each nucleated cell of the body, except eggs and sperms, contains 46 chromosomes. This set of chromosomes is called the diploid because the chromosomes occur in pairs, 22 matched or homologous pairs of autosomes plus one pair of sex chromosomes. The amount of DNA in a diploid cell is written 2n, indicating duplicated chromosomes. The 22 pairs of autosomal chromosomes in our cells direct development of the human body form and of variable characteristics such as hair color and blood type. The two sex chromosomes, designated as either X or Y, contains genes that direct development of internal and external sex organs. The X chromosome is larger than the Y chromosome and includes many genes that are missing from the Y chromosome. The sex chromosome a person inherits determine the genetic sex of an individual. Genetic females are XX and genetic males are XY. Females inherit one X chromosome from each parent, while males inherit a Y chromosome from their father and an X chromosome from their mother. The Y chromosome is essential for development of the male reproductive organs. If sex chromosomes are abnormally distributed at fertilization, the presence or absence of a Y chromosome determines whether development proceeds along male or female lines. The presence of a Y chromosome means the embryo will become male, even if the zygote also has multiple X chromosomes. For instance, an XXY zygote will become a male. A zygote that only inherits a Y chromosome will die because the larger X chromosome contains essential genes that are missing from the Y chromosome. In the absence of a Y chromosome, an embryo will develop into a female. For this reason, a zygote that only gets one X chromosome will develop into a female. Two X chromosomes are needed, though, for normal female reproduction function, however. The sex of an early embryo is difficult to determine because reproductive structures do not begin to differentiate until the 7th week of development. Before differentiation, the embryonic tissues are considered bipotential because they cannot morphologically identify as a male or a female. So what directs some single-cell zygotes to become male and others to become females? So under the influence of appropriate development of signal, the medulla will develop into a testes. In the absence of that signal, the cortex will differentiate into ovarian tissue. So sex determination depends on the presence or absence of the sex-determining region of the Y chromosome, like I've discussed before. So the Y chromosome is also called the SRY gene. And so in the presence of a functional SRY gene, the bipotential gonads develop into testes. In the absence of the SRY gene, and under the direction of multiple female-specific genes, the gonads develop into ovaries. So as you can see in this figure, this is the sex-determining region of Y chromosome, SRY gene, which produces the testes-determining SRY protein and goes into multiple proteins to develop into a testes. Male embryonic development. The SRY gene produces a protein, testes-determining factor, or TDF, that binds to DNA and activates additional genes, including SOX9 and WT1. Please excuse that phone ringing in the background. It also produces SF1. The protein-produced products of these and other genes direct development of the gonadal medulla into testes. Note that testicular development does not require male sex hormones such as testosterone. The developing embryo cannot secrete testosterone until after the gonads differentiate into testes. Once the testes differentiate, they begin to secrete 3 hormones that influence development of the male internal and external genitalia. Testicular Sertoli cells secrete glycoprotein anti-Müllerian hormone, or AMH. Testicular interstitial Leydig cells secrete androgens, or andro meaning male, and then testosterone and its derivative DHT are the dominant steroid hormones in males. Both bind to the same androgen receptor, but the two ligands elicit different responses. In the fetus, anti-Müllerian hormone causes the embryonic Müllerian ducts to regress. Testosterone converts the Wolffian ducts into male accessory structures, epididymis, vas deferens, and seminal vesicle. Later in fetal development, testosterone controls migration from the testes from the abdomen into the scrotum or scrotal sac. The remaining male sex characteristics, such as differentiation of external genitalia, are controlled primarily by DHT. For female embryonic development, the female embryos which have no SRY gene, as discussed before, the cortex of the bipotential gonad develops into ovarian tissue. Research indicates that female development is more complex than originally thought, with multiple genes required for the development of functional ovaries. In the absence of testicular AMH, the Mullerian ducts develop into the upper portion of the vagina, uterus, and fallopian tubes. Fallopian tubes are also called uterine tubes or oviducts. Without testosterone, the Wolffian ducts degenerate, and without DHT, the external genitalia take on female characteristics. So in this picture, you can see that the zygote from XX, which is the female in the bottom, and XY on top for the male, has SRY gene, and that produces into the testes, which then further produces testosterone. And the bottom one is XX, which it has the no SRY gene, and that turns it into the ovaries, and then ovaries produce estrogen and progesterone. Hormone influence. So exposure of non-genital tissues to testosterone during embryonic development is known to have masculinizing effects, such as altering the brain's responsiveness to certain hormones. One controversial aspect of the masculinizing effects of testosterone is its influence on human sexual behavior and gender identity. It is well documented that in many non-human mammals, adult sexual behavior depends on the absence or presence of testosterone during critical periods of brain development. However, a similar cause and effect relationship has never been proved in humans. In human behavior, it is very difficult to separate biological influences from environmental factors, and it will probably be years before this question is resolved. At birth, the infants with pseudo-hermaphroditism appear to be female and are raised as such. However, at puberty, the testes again begin to secrete testosterone, causing masculinization of the external genitalia, pubic hair growth, and deepening voice. By studying the 5A reductase defect in these individuals, scientists have been able to separate the effects of testosterone from those of DHT. And that is the end of my presentation, and these are my references. Thank you.